It's a real privilege to be able to be invited to speak to you today. So thank you for that. And some of the work that I'm going to be talking about goes back to my time when I used to be at the University of Bristol. And so knew Ray from that time there. I used to pop down the road every so often from the university to visit him at the museum. So as Ray said, I'm going to be bringing some aspects of detailed science, particularly through volunteer involvement, citizen science around the horse chestnut leaf miner. And then I'm going to broaden it a bit to bring it up to date for some of the work that I'm doing, which is actually thinking a lot more about people and people within this recording system. I'm sure that much of what I talk about and the principles that I talk about are not going to be particularly new to you. And already I'm sort of aware that as I look through the list of names and recognize wonderful, experienced people, I wonder how much you're going to learn, certainly about the horse chestnut leaf miner, but hopefully a few things will be interesting to you. I'm also aware that citizen science is a bit of a loaded term, particularly within the communities like yourselves, of people who have huge expertise in recording, doing it voluntarily, but huge expertise. And I think citizen science can be used as a term can be used in different ways. So some of which is simply to define that mass participation, beginners getting involved, which is wonderful and is incredible. Citizen science can also be used in a much looser, much broader sense to include the sorts of activities and the expertise which people like you bring. So I hope you will, if you dislike the more broader use of that term, I hope you'll forgive me nonetheless and accept me. I just wanted to obviously start on this fact, and most of this, of course, you will know already, but volunteer collective biodiversity is incredibly valuable. And whether you're looking at the global scale, so some of the UN programs through to the EU programs and the local UK-based programs and so on and so on, it's a really crucial, or citizen science is a really crucial part of this monitoring system that we've got to look at changes in the natural world and in biodiversity in particular. So professionals are another thing, and sensors. So whether that's in-field sensors, which could be collecting on the basis of doing DNA analysis or whatever it might be, or remote sensing from satellites. Citizen science is yet another key pillar in this. And it's great because it benefits science in terms of the monitoring. And so the diagrams there, the graphs there show the trends from the butterfly monitoring scheme data separated into specialist and generalist species. And this sort of data can be reported on thousands of species in reports like the State of Nature. It benefits people in terms of nature connectedness and well-being. I know we all know this, but maybe that's a story that needs to be told a bit more. And later on, I'll be talking about some of the evidence that I've collected in order to look at that. And it benefits actions, because so often as we connect with nature and as we record about nature, it reveals some of the concerns and leads on to beneficial action. Within UKCH, I'm based at the Biological Record Centre. Not as a long and illustrious history as you, but still pretty long. And we're coming up to 58 years in existence and supporting volunteer recorders, particularly through tools for recording. And as Ray mentioned earlier, things like the iRecord system. So over 50 years now, supporting recording schemes right across the tree of life, with 80 or so recording schemes, which we will support or collaborate with at various different levels and degrees, and supporting some of these structured recording schemes as well, where people go out and they follow a set protocol, including well-known, well-established ones like the butterflies, but also some of the newer ones like the plant monitoring scheme, which is just at the stage of producing its first indicators for use by DEFRA about changes in plants and habitats, and the pollinator monitoring scheme, a brand new scheme which has been under development over the past few years, looking to engage more and more people in recording the changes in abundance of pollinators. And as I said, supporting the iRecord recording scheme. So with nearly 100,000 users per year, 20 million records, I think we're approaching the 2 million records per year of a vast number of species, again, from right across the tree of life. So that's where I come from. Now, I want to go back a bit in time, at least in terms of my history, and think a bit, or talk to you a bit about Cameraria ohridella, so the horse chestnut leaf miner. And in particular, this citizen science project, which I ran with a colleague, and to draw people into the recording of this particular moth. And this project all came about when I was a researcher at the University of Bristol. Myself and my colleague, Darren Evans, had been working on projects which were looking at networks. So these big food webs, and actually trying to go out on a single farm and quantify all the, well, not all, a large number of relationships and dependencies between pairs of species. So in total, we recorded almost 1,000, there's about six or 700 species, about 1,000 relationships between those species, able to build this big complex network. And we thought, well, that's all very well. It will get published in scientific papers. But actually, what's the point of us doing this if nobody really knows about it? And so we produced this poster as part of something that we did, and we took to the shopping centre in the centre of Bristol, talking to people about, look, nature is big, nature is complex. We're part of this interconnected web of life. People are part of this too. We're not separate from nature, we're part of nature. So that's why we need to care so much about nature. And so we did this, we took some games, biodiversity Jenga, so children, if they take enough of these blocks out, the tower with the doll on top falls over, and hopefully got across some of the communication about the importance of nature. As I said, the fact that we're part of nature, not separate from nature. And as part of this, we also gave out these little tubes with a leaf miner within it, asking people to rear it and have a look at the parasitoids, which may come out from the leaf. And that was my first real steps into actually doing real, in this case, experimental science, I suppose, with members of the public. And I was like, this is interesting. Let's see how far it can go. And so Darren and I then developed this, looking at the Cameraria, horse chestnut leaf miner. And as I said, we really wanted to not simply do a project where people go out and record, but we wanted to draw people into testing hypotheses, understanding the sorts of ways in which scientists do stuff and the way in which they do it. So there was some element of observation, going out into the field, the parks, gardens, all the rest of it, looking at your horse chestnut trees, but also something experimental. And I'll explain more about that in a moment. I think one of the cool things about this is we did this on a pretty small grant. And we could have paid some field assistants to go across the country and do it, and just sort of travel around the whole country. But being able to do it through this citizen science enabled us to draw a much larger number of people into the project, and actually kept it running for several years, which wouldn't have been possible otherwise. So horse chestnut trees, wonderful, beautiful trees, widely planted in parks and gardens. And if you see specimens like the two I've got at the top there, one next to the Thames and one in front of one of the colleges in Cambridge, you can understand why they're so special and so valued. Of course, in May, you've got the candles of white flowers attractive to bees and so on, which just look absolutely stunning. And I think it's still the case, even though young people are losing some of their connection with nature and feel less connected. Nonetheless, I still think for many, many children, if you ask them where to go and collect conkers locally to them, they will be able to take you to the best place to collect conkers. These beautiful, shiny fruits from the horse chestnut tree. And then people collect them. Allegedly not so good for spiders, if you're interested in house spiders in your house, because they're supposed to stop the spiders. But I think that remains scientifically untested. Maybe there's a citizen science project in there somewhere. But of course, now what was it, more than 15 years ago now, we started seeing blotches on our horse chestnut trees, these white leaf mines. And so for many people, this was quite possibly the first time that they had really experienced and come across leaf mines. And of course, a few blotches on a horse chestnut leaf is just one of those things. But you can see as you go around the pictures, I think most of these pictures were taken around the end of July. You can see actually in the worst cases, you can get the trees, certainly at ground level, appearing like they're decimated. Almost all the green from those leaves has been consumed by the leaf miners. And each one of those leaf mines is maybe about half the size of a little finger. And so that devastation for the horse chestnut leaf is simply due to the sheer number of leaf mines and leaf miners within that leaf. So it's, I'm sure many of you are familiar with this moth, but it's brilliant to be able to talk to people about this, people who don't know much about moths and sort of think about them as brown and boring. You've got this beautiful little bronze moth with black and white stripes about the size of a grain of rice. And then it's got these caterpillars. I'll just play this little video here. These caterpillars, which are adapted for living between the top and bottom surfaces of the leaf. And you can just see there, it's got this anvil-shaped head. It's dorso-ventrally flattened. And if you open up one of these, you can really see the adaptations of that leaf miner or that larva to living and existing within the leaf. And just to note there, it was described in 1986, and I'll come back to that in just a moment. So the horse chestnut leaf miners produce, usually it's 3 generations of adults per year, sometimes a partial 4th generation at the very end of the year, depending on where you are in the country. And it's, of course, the larvae which do the damage to the horse chestnut trees. And it can be quite stunning because particularly in that first generation, it's quite a synchronized emergence of the moths from the pupae and the leaf litter underneath. And over the course of a few days, round about the middle of May, it can look like the tree's almost shimmering in the morning with the thousands upon thousands of moths emerging and then mating, laying their eggs. So I said that this moth was described in 1986. It was first found as brand new to science in the early 80s in the Balkan regions. And you can see there the pretty rapid spread across Europe. And so that map goes up to 2017. It's basically found anywhere in Europe now where there's a horse chestnut tree, and having recently got to Scotland as well. And so this provided this real conundrum. So you've got this horse chestnut leaf miner existing on the horse chestnut leaves, suddenly from the Balkans, where the horse chestnut is a native, spreading rapidly through Europe, producing these really large populations. It's acting like the sort of classic invasive species with these huge spikes of populations. Never been seen previously to the 1980s. It's the only species of Cameraria in Europe. There are other Camerarias, which are leaf miners of Acer in America and South Asia. So that suggests that it was introduced to the Balkans from some other part of the world. And from there, it then spread from the native trees into the planted trees and spread across Europe. So that seems the most natural explanation. But as people looked at this, they discovered actually, when you go to the native stands of Aeschylus, the horse chestnut tree, there's really high genetic diversity of these moths. Well, that is an indicator of them being native and established there for a long time. There's also really high parasitoid diversity in these native stands. Once again, it makes you think that it's a native and has been a long-standing native in that region. How come no one's ever seen it before? And so I mentioned this study just because I think it's the most brilliant bit of detective work. And this is, in this study, they went out and they sampled the horse chestnut leaf miner from places where it was native and got those two findings about the genetic and the parasitoid diversity. And they were really wondering and found it a real conundrum, the fact that this species appears to be native, and yet had never been seen previously. And so they started going to look at herbarium specimens. And eventually, after a lot of hunting around, they found this herbarium specimen of a native horse chestnut leaf, which was collected from Greece in the late 1800s. And if you know anything about the horse chestnut leaf miner, you will look at that little inset and think that looks pretty familiar. They did the genetics on it, and yes, indeed, it did turn out to be Cameraria. And in fact, what they found was that as they were looking at various herbarium specimens, they did find a few instances where the fingers, if you like, of the leaf and the horse chestnut leaf in a few places were sort of folded over slightly over each other in a slightly strange configuration. And of course, looking under that, they then found one of these leaf mines. Of course, what botanists at that time were doing were not collecting simply to understand the biodiversity of the region. They were there to collect the plants of the region, and so would pick the most pristine leaves. And clearly this, from probably a botanist at the time, this might be seen as a bit of a failure to have a leaf pressed with a leaf mine within it. So some really fascinating detective work, which I think shows the value of herbarium specimens and museums and all the rest of it, keeping this archive of biodiversity in the past. Right, I could waffle on about that for ages, but I won't. So the horse chestnut leaf miner, discovered as new science, well, actually in the sort of early 80s, late 70s, reached London in 2002, and it spread at the rate of about 30 kilometres per year. So when we started this project about 2012, you can see that it had got to the Midlands and beginning to spread up through Yorkshire, but was only been established in those places a few years. In London, it had been established there for a decade. And so this is the project. So we created this web page and asked people to record asked people to record two things about the moth. So the first one was we set people these two missions. The first one was about is the level of damage to the trees greatest where the moth has been longest? So we asked people to go out, have a look at their trees and score the damage to the leaves according to these simple categories. And you can see that we got a really nice spread of records. It's about 2,000 records from across the country. But the point is that constantly we refer people back to this question. We're not simply getting, hey, this is the distribution of the moth, but trying to understand where the damage had been caused. Now, I've done teaching on citizen science on a few different university courses and things like this. And I always feel very old when I say that actually we developed an app as part of this. Gosh, smartphone app, can you believe it? Of course, nowadays, smartphone apps, you would do that for almost any project, nearly. At the time, this was one of the first biodiversity recording apps, certainly focused biodiversity recording apps of its kind. And it was from the technology of this that actually several apps were built upon this and then slowly developed. But we spent a lot of money actually really designing this in terms of making sure the usability was really great. And it clearly worked. It got tweeted by Stephen Fry and made the news simply because we were a project with an app, which is bizarre to think about it 10 years on. So the question, does damage increase with the length of time Cameraria has been present? We're able to take the records, do what ended up to be some fairly sophisticated statistical analysis. And you can see those are the categories 1 to 4 up the side. And we looked at how long Cameraria had been present in a location and the level of damage to the leaves. So what you can see there is that it takes 3 to 4 years to reach the maximum levels of damage, after which it appears to be stable. Now that's something which had been known anecdotally, but never actually quantified in a scientific sense. So this project with 2,000 people from across the country taking part did produce some It's not utterly groundbreaking, but it was some novel scientific results. And I think it was great to be able to draw people in on this journey of actually doing the science, not simply making records. So there are some pictures of the horse chestnut leaf miner, and I should credit Rich Andrews for this picture. If you rear the leaf miner, there are two possible outcomes. Either you get this alien moth, so-called alien moth, or you get the natural pest controller. I should say, actually, some of the conversations around this so-called alien moth were really interesting because, of course, the horse chestnut tree is not native to Britain. It's only widely been planted for the past 200 years anyway. And so you've got this non-native moth on a non-native tree. And one of the other fascinating things, I think, is it allowed us to ask questions and have conversations about values within conservation. There aren't these really clear rights and wrongs and shoulds and shouldn'ts within conservation. It's very much values driven. It's about people and their interaction with nature. So that was interesting. That was good to be able to discuss and talk about that. So the second mission we set people was the pest controllers mission. So are the levels of pest control, so assessed as the rate of parasitoids versus the moths which were reared, are they greatest where the moth has been longest? The great thing about this is that at the right time of year, once all the moths have pupated in the leaf, then you can pick a leaf, stick it in a Ziploc plastic bag, wait for 2 weeks and count and see what's come out. So a brilliant opportunity to engage school children and talk to them about the amazing life histories of parasitoids being eaten from, or eating their prey from the inside out and then bursting out, and all this sort of gory stuff which just amazes the children. Also allowed us to dig into a bit of the natural history of these parasitoids as well. And we wrote an article in British Wildlife, and keys available on our website as well if you dig around there. Because there's a remarkable diversity of parasitoids which prey upon Cameraria. They're recruited from the fairly generalist parasitoid community that feeds or prey upon other leaf miners. But it also it's about 25 or so species which are known to parasitize horse chestnut leaf miner. And so it becomes quite a doable thing that if you are a bit more expert, particularly if you've got a microscope, actually have a bash at identifying them. Maybe parasitoids have always been something that is just like, that's a group that's way too complicated. Well, you can begin to do it with this constrained community, which I think is interesting. You can see the size of these things, though, and just once again communicating with people about the absolute beauty, incredibleness of nature, and these small things which drive drive food chains and communities, and are really important. So we got a spread of records from around the country, working with school children and putting it out for people to publicly take part. And you can see that the parasitism, once again, once we do the analysis, parasitism increased with the length of time that the horse chestnut leaf miner had been present, with certainly we've extrapolated up from 2005 backwards, but you were getting rates of 5–10% parasitism, which actually people elsewhere in Europe have done the work to show that that's nowhere near sufficient to moderate the numbers of the horse chestnut leaf miner in places where it's established. So all of this work was published, and one of the great things actually was that, so you can go back and you can say, well, how on earth can you trust that children can count moths and parasitoids correctly? And the way in which we did it was we went and we checked, we ground truthed their observations, and what we found was they can't, by and large, count parasitoids very accurately, but they do count them wrongly in a predictable way. And so we were able to take account that error rate within our analysis. So we did the analysis taking that known error rate into account in order to come up with this result. So we engaged lots of these school children, and we got them to ask, or we got them to answer how they found the project. And you can see there, I always like to point out, if you look sort of towards the top, there's a tiny little boring, but clearly they really enjoyed it and found it great fun and interesting. So for me, I think that's a real positive thing. I'm going to go back to this thing about fun and enjoyment a bit later on in the talk, but I've got something else that I wanted to talk about first. And this is this project called the Decide Project, which I've been running for the past couple of years. And like the projects that I talked about, the Conquer Tree Science, and many other projects actually allow people to take part in biological recording wherever they choose. And that's fabulous. It really opens things up to many people. But, and I'm sure this isn't news to you, what that leads to is spatial unevenness. So both at large scale, get loads more records in southern England than you do in Scotland or the Midlands of Wales. And at small scale, you get more records at the edge of towns than right in the countryside, in the agricultural areas or in the town centres. And so you get this spatial unevenness, which can be a real challenge in terms of dealing with the data that we've got. And this led us to this sort of tagline around the project, which is that all records are valuable. Maybe you could qualify that a little bit, but by and large, all records are valuable. But we don't simply need more records, we need more informative records. So if you were a recorder, how do you choose where to go in order to add most value to your records? Now, for many of you here, you will be pretty expert in terms of recording, and I'm sure you, as you go around, you've got your own particular plan of sites that you would like to cover or places that you would like to go. You might be doing it at a countrywide mapping or whatever it might be. But for many ordinary people, having an understanding of where their records are going to be most valuable is really important. So one of the ways in which we've been exploring this is with the targeting revisits maps. And if you go to the BRC website or do a search for this, you'll be able to find out a bit more about this. So we've created these interactive maps, which are updated in real time for a range of different taxonomic groups which are traditionally not brilliantly well recorded. And you can see some of the maps there. It's got the distribution of points, and you can see lots of gaps in those distributions. But you can also see that they're color-coded. And so the green squares are squares where we've got records in more than 1 year. And the pink squares are squares where we've got records in only 1 year in the past. So why is that important? Well, we can do analysis, and we've got I've got colleagues who do a lot of analysis of these type of data, so-called unstructured data, and we use a form of analysis called occupancy modelling to be able to use these data and produce trends of changes in occupancy, which is sort of like changes in the overall distribution over time. And we can produce these trends for a couple of thousand species now, which is absolutely superb. But the only data points that go into those models are data points from sites, in this case 1 kilometre squares, where we've got records in more than 1 year. So if we've got records in 1 year in the past only, if I went back to that square nowadays and made another record, then both the historic record and my current records would go into the data set for occupancy modelling, which is brilliant, feels like a real win-win for the use of the data and also for me as a recorder. And so we set people this really simple challenge of, can you make the pink squares green by going and recording, in this case, the communities of grasshoppers or the communities of soldier flies or whatever it might be. And it provides that bit of targeting and that bit of information for people to really think about where, according to this simple criterion, where their records are going to be most useful. We're currently inviting people to use, explore and give feedback on these tools. So the links are on the targeting revisits maps and also on the BRC news item about that. So I'd really welcome you and your involvement in giving feedback on this. And feedback so far has been really quite positive, and then thinking about how we can build it into tools like iRecord, because currently we're taking data from iRecord, so updating these records in real time from that data, and then at the end of the year we'll do a sort of batch update with all the records that have come in from other sources as well into these recording schemes. So I'd encourage you to visit that. I'm also going to be running a focus group or two over the coming month or so. So if you were really keen on getting involved with exploring how this might be improved or the value it is, then please do drop me an email, and it'd be brilliant to have people being involved in those. Within this Decide project, we spoke to a range of different people who use data. Now, of course, people who use data include recorders like yourself, keen recorders, conservationists, and so on. But we were talking to people in policy, decision makers, those sorts of people. And they acknowledged that we have good information for many species to do this large-scale reporting of trends in abundance or distribution. And also for particular sites, nature reserves and things like that, in many cases, we've got pretty good records that allow us to manage sites well. But looking at something like that picture on the right, that map on the right of the distribution of smooth newts in London, it's really patchy. Well, is that patchiness because there aren't newts or because there aren't recorders? Of course, this is one of the perennial problems with dealing with these sorts of data. So those data users were saying, actually, maps like this, dot maps like this are great, but really limited because I don't quite know how to best use the data. We need these comprehensive maps. Ideally, we need them at fine scale because a lot can happen within a 1 kilometre square. There can be a lot of variation. And if it's people, a regional economic forum or a partnership or something like that, let's trust that they want to make good decisions for both for the economics and for jobs and so on, but also for nature, as many of them genuinely do. Well, they need this fine scale information in order to be able to balance these decisions well. So they need comprehensive information. Now there are, I think it's 25 million hectares in Britain. So that's a lot of squares if we were going to simply go out and record in every square. Recorders, I go back to the slide earlier about the targeting revisits maps, recorders lack these tools to guide their recording. People were saying, I want to record where I choose to record, but sometimes, you know, I would like to record in places where I know the records are going to be really valuable. So how do we bring those two together? So we did this, worked with a bunch of different partners. You can see their logos on the left there. So we used data of butterfly and moth recording. So records from over 100,000 recorders over 20 years. So we've got these 18 million records, species records, absolutely incredible. But if you plotted each species on a map, still look a bit patchy, particularly at the 1 kilometre scale. And so we created these models, go on to talk about them in just a moment, but produced models of the distributions, so the comprehensive fine scale distribution of 800 species. And then the key thing about this particular project is we then joined it back up because we fed the results of those models back to the observers, the recorders, allowing them, and we did this in a way that was accessible and relevant to them, allowing them to spatially target their recording effort to choose where to go next. Excuse me. So with these species distribution models, we took the dot maps of records of species, and we also took 33 environmental layers, most of which have been derived from satellite information. And the satellites obviously are superb in terms of giving us high quality information on certain attributes of the land, combined them together, and we were able to create this predicted distribution for any particular species. But each of those distributions would have a probability of the species being present, but would also have an uncertainty around that. And so you can see that on the map, there are certain places where the maps are particularly uncertain. And so we could then say, well, let's encourage people, or let's provide tools so that people know where these distribution maps are most uncertain. Those could be the places which are most important for people to go to. And so we created this Decide tool. The messages were, if you want to discover new places to record wildlife, then you can explore the tool. It shows these places of high priority for recording, or higher priority for recording. Of course, all records count, all records are valuable. We kept communicating that, but these are places where maybe on this particular basis, there's particular value for us to get their records from there. And then, of course, you can use the existing tools, whichever route of recording that you prefer, you can do that. And so we created this tool, and you can go on and you can explore it yourself. So for any particular 5 kilometre radius circle, you can look at the colours, which are the recording priorities. You can click a button and it will give you suggestions based on the information that we have on publicly accessible places, so footpaths, areas of open access land and things like this. This tool is all updated in real time from iRecord, and you can get a whole load of extra information about any particular site on that map by clicking on it. So you can look to see whether it's the fact that there haven't been records made there recently, or it's the fact that model uncertainty is high, or whatever it might be, and it gives you some ideas of what to expect. And over the course of the past year, we've had nearly 2,000 people get involved in using the tool. And actually, people have been spending on average nearly 4 minutes on this tool. So really exploring it and using it. And clearly many people coming back again. And thinking of the targeting revisits and the Decide tool, people have given quotes like this: being able to see how impactful my recording can be and directing me to where recording could have the greatest impact is immensely powerful and motivating. And we wanted to sort of up really that sort of impact and that engagement with people as we were going along. And so we gave people the opportunity last year to take part in this experiment where they signed up for weekly data stories about the impact of their recording, in this case, butterfly recording. And so we gave people, randomly, we assigned people different types of data story, or different emails to receive. So one week you might receive some maps telling you about where you've recorded, but where are the high priority places that you could record next. Sometimes it was simply a photograph, sometimes it was almost like a league table showing you how you're doing compared to others. And so trying to explore this idea of are these nudges, are these sort of feedbacks? Should we emphasize competitiveness? Should we emphasize collaboration? And what we found was that people really loved getting these. They really appreciated them. Actually, from the analysis that we've been doing so far, it looks like those people who received these did more recording than would have been expected than those people who didn't receive these emails. So simply getting those prompts seems to be really, really valuable. So feedback to recorders is vital. And of course, we're quite community-based. In biological recording, quite community-based. Maybe we sort of have that implicit competition, but we don't like to acknowledge it often quite too strongly. But really interestingly, it was the emails that we set up to be competitive and to play to people's competitive side, which were the ones which actually people really liked and responded to most, which was fascinating, because when we were initially talking about this within some of the focus groups, people were saying, no, we don't want to be competitive. That's going to put people off. So really fascinating. And in fact, what I must recognize is all of this was done through a great process of co-design, getting feedback from people, engaging keen recorders through to beginner recorders in focus groups. And I noticed a couple of people involved in the focus groups were a part of this meeting. So just to say huge thank you to you for your time and your feedback. It really was brilliant to be able to help us design something that's a tool So I'm a recorder myself, so it's sort of designed by recorders for recorders, not designed by scientists to tell recorders what to do, but really trying to align these motivations so that, yeah, we're working with recorders for recorders. We also, just as another little thing, we gave out a whole load of these LED bucket traps for moths, which are brilliant because they run off a mobile phone power pack. You don't need generators, you don't need mains. The catches are not quite as good as you might expect from an actinic bulb or anything like that. But for something that's just left out and about, obviously with landowners' permission, a really great tool that I think increases our opportunity to do moth recording, nocturnal moth recording, in a wider range of places. Right, final stage, last few minutes. I just wanted to talk about people. So we talked about people and recorders and how I might engage and think about those recorders. A couple of years ago, actually just at the end of the first set of the strictest lockdowns, I ran this project with colleagues, including a couple of psychologists, talking about a wellbeing experiment, thinking about the benefits that people get from engaging with nature. So it's known that being in nature makes people happy. During the lockdown, people engaged a lot more with everyday nature. It's more important than ever to their well-being. So the lockdown was something where people seemed, when they could switch off from a lot of other things or needed somewhere to de-stress, nature was absolutely vital to them. So we created this project, which was a randomized controlled trial looking at the impact of nature-based citizen science on wellbeing and nature connectedness. And we had 500 participants getting involved with this project, and the paper has just been published. So you can read all about it in the journal People and Nature. It's open access, so anyone can get access to it. So what we did was we recruited people and we did a pre-survey on their nature connectedness and measures of well-being and things like this. We then asked them to, over the course of 1 week, to get involved in a nature-based activity 5 times. After they'd done that, we then gave them another survey about their nature connectedness, their happiness, their well-being, things like that. This is all a very typical environmental psychology sort of experimental intervention approach. So the different groups that we put people in was either doing nothing. We just said, okay, we'll get back to you in a week. And so they were the control group, or they did citizen science, which was either we invited some people to simply do some butterfly recording using the iRecord Butterflies app, or we asked them to do one of the FIT counts, the flower insect time counts, for 10 minutes in their garden or space where they were able to get to. Or we asked them to do this noticing 3 good things in nature. So spend 10 minutes in nature, just use your senses, experience nature, dwell on how nature makes you feel, how connected you feel to nature, these sorts of things, and write down 3 good things. And then we also did this combination. So while you're recording your butterflies, while you're recording your insects, take a moment or 2 and also notice the good things in nature and write them down. So those are the 3 groups. And remember, there was that control group as well. So when we looked at these psychometric scores, what you can see, this is the comparison of citizen science, 3 good things in nature, or the combined effect compared with the control. And you can see that in all cases, they are, well, in almost all cases, they're higher. So getting involved and engaging with nature gives benefits to nature connectedness, which are those first 2 sets of bars, or happiness, your satisfaction with life. And actually, even then asking people about their behaviours and their conservation behaviours, it also had an impact on this. Just 10 minutes a day for up to 5 days a week in 1 week had this impact. So these benefits are known in terms of people connecting with nature, but it hadn't been quantified for citizen science, doing this biological recording. I'm sure every one of you knows actually the joy and the benefits that you can get from engaging with nature through biological recording and all that interesting challenge in terms of learning and all these other benefits as well, and being part of a community. But there we go, scientifically proven, taking part in nature-based citizen science is good for you. And my colleague Miles Richardson from the University of Derby, he's produced this Nature Connection Handbook, which talks about research that he's done on these 5 pathways to nature connection. And we explored these with those people who took part. So it's about using the senses, not just sight, but all of your senses, seeking and appreciating the beauty of the natural world, noticing and thinking about the emotions, maybe the calmness or the joy that it brings, that meaning, how nature gives you that sense of meaning and what it means, things like seasonality and all sorts of things. And then that final pathway is about compassion, helping and caring for nature. And actually what you can see is that these different activities, 3 good things in nature and citizen science in particular, just all connected people with these pathways to nature connection, but they did so in slightly different ways. So those doing citizen science really felt more like they were helping and caring for nature. Whereas those doing the 3 good things, we asked them to use their senses, and sure enough, they really responded positively to that. The interesting thing is going out, doing this sort of citizen science, the 10 minutes at a block of nature-based citizen science, and asking people to also actively notice nature had consistently the most positive effect. And I think for me, that's really exciting because it opens up the opportunities about where we can go in terms of doing citizen science, which actually intentionally boosts people's connection with nature and all the benefits that can bring. And finally, the final slide is thinking all about that, in a sense, that slightly softer stuff of connecting with nature. I think we're really, as a community, we're really aware of all of the things like the beauty of nature writing and the connection that can bring, as well as all the science that can bring so much joy and interest and inspiration to us. So as part of the DECIDE project, we were exploring about this question about what happens when we turn nature into data. How do we go from this sort of soft experience, if you like, that emotional experience of me going out, recording nature, seeing that hoverfly as I did this morning on my walk, getting that sense of joy, that connection, all the rest of it, making it a record, at which point it becomes digitized and all of that emotion is stripped out of that record. So what does that mean? So we were working with a couple of artists, a painter and a poet, to explore this and ran this immersive activity that people could listen to a 20-minute poem as they explored the woods at night, coming across these illuminated caskets, thinking about if data could dream, then what would it say in this time of declining butterflies and moths? Which I think hopefully is taking you all the way through from this amazing hypothesis-led science that people can do through all of the ways in which people can get involved in recording, the benefits that people can get, and these sort of added benefits of people engaging not in a scientific way about the data, but just people who took part in this were really blown away. I was blown away by their positive reaction to wandering around at night, experiencing nature in this way, experiencing the art, the poem, and really getting something from it. So I will leave it there. Thank you so much. I hope at least some of those bits have been of interest to you, and I would welcome any questions.